Good morning, everyone. Welcome. Thank you for coming. Today we have with us Dr. Oliver Press. Dr. Press completed his Bachelor of Science in Biology at Stanford, his medical degree and PhD here at the University of Washington, after which he traveled to Boston and completed his internship and residency. He then returned to the UW for his chief residency and completed his fellowship in oncology. Dr. Press is now Professor of Medicine, Adjunct Professor of Bioengineering, and recipient of the Dr. Penny Peterson Memorial Chair for Lymphoma Research. He also serves as the Chairman of the Follicular Lymphoma Consortium Executive Committee of the Lymphoma Research Foundation, Vice Chair of the American Society of Hematology's Committee on Lymphoma Neoplasia, and Co-Chair of the National Cancer Institute's Steering Committee for Lymphoma Research. His awards are numerous and include our Robert S. Evans Award, election to the American Society for Clinical Investigation and the American Association of Physicians, and the Frederick Leadership Award from the Lymphoma Research Foundation. Dr. Press is quite busy. He's had well over 360 publications. He's got a number of ongoing phase 1, 2, and 3 trials, and continues to be quite active in mentorship with both medical oncology fellows as well as postdocs. But he's taken a bit of time out of his schedule, and we thank him for presenting Grand Rounds today, Emerging Immunotherapies for the Treatment of B-cell malignancies.
Thank you for that very kind introduction. I'm happy to be here and to talk a little bit about the exciting developments in the field of immunotherapy for hematologic malignancies. I'm told that I need to begin by declaring my conflicts of interest, and I do have a few shares of stock in Emergent Biosolutions. I'm doing some clinical trials for which the patient care expenses are reimbursed by Genentech and Roche. I've taken an honorarium for giving a CME lecture on lymphomas, and most of the things I'm going to talk about are new and exciting drugs and treatments which are not yet approved by the FDA. The disease we're going to talk about today mainly is non-Hodgkin's lymphoma, and many of you may know that this disease has almost quadrupled in incidence over the last 60 years. Very impressive rise. I'm happy to point out that the mortality curve is diverging from the incidence curve, and this improvement in outcome of patients is largely attributable to the introduction of effective immunotherapy, mainly the monoclonal antibody Rituximab. The diseases I'm going to talk about today are listed here. There are 67 different types of non-Hodgkin's lymphoma, which is way too much for a lecture like this. I'm going to simplify the world and lump the lymphomas into either the slow-growing, low-grade indolent lymphomas, or alternatively, those lymphomas that grow very rapidly are called intermediate or high grade, and I'll call those aggressive lymphomas. We'll also talk a little bit about chronic lymphocytic leukemia and ALL. And since most of you are not oncologists, I just wanted to make sure everybody's got the jargon down. When we treat somebody, if we are able to get rid of every vestige of disease by every test we do, we call that a complete remission. If we make a dent on the disease and shrink it by at least 50%, but there's still evidence of some disease remaining, we call that a partial remission. The overall response rate is the addition of a CR and a PR rate. Progression-free survival means that the patient we've treated is not only alive, but there's no evidence of disease growth. And overall survival is self-explanatory. That's whether the patient is alive or dead. The treatments that are used for the B-cell malignancies are listed here. Surgery is really a diagnostic test for most of our diseases, not really a treatment. Chemotherapy remains the mainstay, with radiation used for localized disease. But more and more, we're focusing on the use of immunologic interventions to treat these patients. Most prominent, of course, is the use of monoclonal antibodies, and I will talk about those. I will also speak about radioimmunotherapies, antibody-drug conjugates, and adoptive T-cell immunotherapy. I don't really have time to cover the vaccines, and there aren't as many new developments in that area in lymphomas anyway, so I'll have to defer that to some other time. I remember one of the first grand rounds I ever attended in this room was by Alex Pfeffer, and he put this slide up, and I think it applies as much to everything I'm talking about today. A new treatment comes out, it looks promising, everybody gets excited, there's an article in the New York Times or Time magazine, everybody's sure it's going to be the answer to every disease, and then problems emerge as it's experienced more and more, and then people decide it's absolutely worthless. And the truth is probably somewhere in between. I've seen monoclonal antibodies go through this in the late 70s, early 80s. People were very excited, and then the mid-80s to early 90s, people thought antibodies would never work for any disease. It's hard to get funding for that research, and now, of course, they're a standard part of our cancer therapy. So keep that in mind as I go through some of these new agents. I'm going to begin by talking about monoclonal antibodies. And just remind you all, the structure includes 2 heavy chains, 2 light chains. There is an antibody combining region, actually 2 per antibody, and these undergo somatic hypermutation and bind very specifically to some target antigen. The constant regions are just that constant, but they do have important functions such as fixing complement and attracting effector cells, be that natural killer cells, T cells, macrophages, etc. And there are a panoply of lineage-associated antigens that can be targeted. Most of these antigens are present on B cells, both normal and malignant ones, but not on any other cell in the body. And they're also not on plasma cells, the terminally differentiated B cell, nor on hematopoietic stem cells. And it turns out you can ablate normal B cells with very few problems for at least up to 2 years. And that's because the plasma cells keep churning out antibodies, they're long-lived, and because eventually you can regenerate your B cell pool from the hematopoietic stem cell. And the primary targets so far have been CD20 against Rituximab has been built, but CD19 and CD22 are important. And there have been products, some of which are FDA approved, against many of the antigens that I show here. It's important also to remember the history of antibody development. Initially, this was done by injecting things into mice. And so you had murine antibodies in general, shown here in white. And then as people got smarter in the lab, they engineered more and more human components onto it. So for example, Rituximab is a chimeric antibody where the parts in blue are human and the parts in white are the retained parts that bind to the antigen. You don't want to mess too much with those. And the chimeric antibodies fix complement better, fix human complement better, and interact better with human effector cells than murine antibodies. And then you can get even fancier, humanize it more, and just leave the little complementary determining regions, murine, or there are now tricks by which you can generate entirely 100% human antibodies. And the FDA has given a very confusing nomenclature, which we all have to use, and I'm not going to go through this in detail, but it's there for those of you who are interested. Now, how antibodies work to kill cancer cells remains quite controversial. There are probably many mechanisms that are important. Most authorities in the field think that antibody-dependent cellular cytotoxicity is most important. That is, once an antibody binds to an antigen on the surface of a tumor cell, it engages through its Fc portion effector cells. Those could be NK cells, neutrophils, macrophages, whatever, and brings those cells next to the tumor cell, and then the effector cell kills it. Another mechanism is that the C1q portion of complement can affix to the Fc portion of an antibody attached to a tumor cell, and then this creates pores that lead to cell lysis. Finally, there are direct effects. Just by the fact that an antibody ligates its target, often sends signals through the cells and disrupts intracellular signal transduction pathways, and can either interfere with cell growth or it can lead to programmed cell death or apoptosis. So those are the 3 main mechanisms that people talk about. There are some others, but these are the main ones that people generally analyze. Now, many of you don't know that the first trial ever done with a anti-CD20 antibody in humans was done here in Seattle. I happened to be a fellow at that time, working with Don Thomas and Fred Applebaum and Paul Martin. And we used an antibody called 1F5 that was made by Jeff Ledbetter, who's currently a faculty member here in rheumatology, and Ed Clark, who's a faculty member here in immunology. And we gave this to some patients who had circulating lymphoma cells, what were called lymphosarcoma cells in those days. And as you can see in the bottom line, as soon as we gave the antibody, they virtually disappeared, at least for a few days, and then they came back. Similarly, patients who had pathologically enlarged lymph nodes experienced marked shrinkage of those lymph nodes. This was a wholly murine antibody, and the problem was, shown here, that although we improved the patient's situation temporarily, that was short-lived, a few weeks or a few months, and then they were kind of back into the same kind of situation. And so this led to some think tanks, and some of the ideas were better than others. We actually were visited by a guy named Richard Miller, who at that time was the founder of a company called IDEC down in California. He came up and met with Fred and me and talked about our preliminary results. He also talked to a lot of other people, and it's hard to know how much influence we had. But a year or two later, the company he founded, IDEC, came out with this chimeric antibody called Rituximab, and that kind of has revolutionized the treatment of B-cell malignancies since then. This is the pivotal trial that led to its approval, and at the time it was, I thought, relatively modest results. Used as a single agent, most of the patients who have a relapsed indolent lymphoma, like a follicular lymphoma, will experience tumor shrinkage, as shown in this waterfall plot. You're going to see a lot of these plots, and basically 0% means the tumor is the same size after treatment that it was when you started. The distance below the line indicates how much the tumor has shrunk, and if it goes above the line, these are patients whose tumor actually grew through the treatment. You can see most people benefited, and about half of them reached what we call an objective remission with at least 50% shrinkage, and a few were lucky enough to get a complete remission. So this is using it as a single agent. However, its greatest benefit turns out to be that it synergizes with chemotherapy. So here's a phase 3 head-to-head comparison of patients with indolent slow-growing lymphomas treated either with Cytoxan, vincristine, and prednisone, or the same regimen along with Rituximab. And you can see the time to treatment failure, that is the time until the disease grew again, quadrupled from 7 months to 27 months. So this is a pretty dramatic finding. It also translated into an overall survival advantage for these patients. Even more impressive to most of us in the lymphoma field were the results in aggressive, rapidly growing lymphomas. This was a phase 3 randomized trial done mainly in France. The regimen called CHOP versus R-CHOP. CHOP was the standard of care in those days in the early 2000s. And what turned out is that you just about doubled the event-free survival, that is the time to either relapse or death, if you used R-CHOP compared to CHOP. Look at this is 4 years, and it's about twice as much. So this was pretty exciting, particularly for people with aggressive lymphomas. And you can see that the overall survival was also improved, not as dramatically, but because there are salvage second and third line treatments, but there were about 15% better survival using the antibody-containing regimen. Well, you might ask that if Rituximab is so great, why are people spending so much time and money to develop a better CD20 antibody? Now, you might say, well, it's a $5 billion a year drug, and people want to get a piece of that action, and so that's part of the reason. But there are still lots of other reasons as well. If you use it by itself, only about half to maybe 70% of patients with indolent lymphoma will respond as a single agent. Only about a third of patients with aggressive lymphomas will respond to it as a single agent. And even if you use Rituxan with chemotherapy, most people with indolent lymphomas eventually recur. It may be many years later, but eventually most of them do. And furthermore, 40% of patients with aggressive lymphoma still die of their disease. So there's lots of room for improvement. Not only that, but it's a difficult drug to give in many ways. It takes 2 to 6 hours to give it because otherwise there are very bad infusion reaction. So it'd be nice to have an antibody that didn't do that. It's also an expensive drug, and so many people are trying to enter the fray, have a little competition, comes off patent in a couple years, and there are a slew of biosimilars that are just waiting. But there are also innovations by molecular engineering which promise to introduce even better antibodies. So the first generation are the murines and the chimeric rituximab. The second generation are the human or humanized CD20 antibodies. And then third generation are those that are not only human or humanized, but also have modifications to the FC region, either to make them bind complement better or to have tighter binding to FC receptors. And there are a zillion of these. I've just put a few of them on this slide because otherwise you wouldn't be able to read the print. And I'm not going to talk about them all, but I'll talk about a couple. The first one I'll talk about is actually FDA approved, and it's an antibody called Ofatumumab. It binds to a different region. This is the CD20 molecule. It's a molecule that winds its way back and forth through the cell membrane 4 times. And the epitope that Ofatumumab binds to is said to be adjacent but not identical to the Rituximab epitope. It's also a little closer to the membrane, and therefore complement seems to work better with this antibody. So it has better complement-dependent cytotoxicity than Rituximab does. It has comparable antibody-dependent cellular cytotoxicity, and it has been shown to have some modest benefits in chronic lymphocytic leukemia. This is a disease which, when it becomes refractory to our standard chemotherapy drug fludarabine and to a CD52 antibody that's used called Alemtuzumab, is very, very difficult to get to respond to anything else. And they did a trial with Ofatumumab showing that about 60% of patients had objective remissions. Also, patients with bulky lymph nodes who were hard to treat with standard therapies got remissions. They only lasted, if you look at the median duration of progression-free survival, it's about 6 months. So it's not, it's not a home run, but it's better than anything else we had at the time, and so it received FDA approval. How it's going to fare in the other diseases other than CLL remains to be seen. There are a slew of phase 3 trials, only 3 of which I put up here. One is a head-to-head comparison to Rituximab in relapsed follicular lymphoma. And then there are trials with either our chemo or Ofatumumab chemo head-to-head. And they're huge trials with thousands of patients being enrolled right now to see if it's better. The single-agent trial in relapsed and refractory follicular lymphoma are not particularly striking, unfortunately. It looks like the overall response rate of 42% is about what you'd expect with Rituximab. And if you look at patients who have grown through Rituximab therapy, only about 11% respond to Ofatumumab. So I think the jury is still out. It does have a role, certainly, in resistant CLL. Whether it's going to displace Rituximab for other indications, I think, remains to be seen. Perhaps one of the most interesting new CD20 antibodies is this one called Blinatumomab, formerly called GA101. And this antibody is different in several ways. First of all, it's been molecularly engineered so that the arms of the antibody are at a funny angle, and this allows it to be much more effective at inducing apoptosis or programmed cell death, just the way it engages the antigen. Also, it has been produced in the laboratory in a way that the fucose residues are diminished in the area that binds to the FC receptor. This results in much tighter binding, particularly to certain allotypes of FC receptor. And so this is another innovation. And so if you look, the GA101 or Obinutuzumab antibody has more ADCC than Rituximab, but much better apoptosis induction and less complement fixation, deliberately because they felt that the complement fixation was mainly responsible for the infusion reactions. That was the theory, and they wanted to engineer it out. In mouse lymphoma xenograft models, it outperforms rituximab substantially. And you can see that with the new antibody, you can cure mice, whereas you couldn't with rituximab at the same dose, and at every dose, GA101 looks better than rituximab. So that part of it looks good. There has been a randomized phase 2 study head-to-head of rituximab versus Blinatumomab, which we participated in, and the preliminary results are out. Again, it's phase 2, so it's not going to be definitive. If you look at the overall response rates, it looks like the GA101 looks a little better than rituximab as assessed by the investigators. There was an independent blinded review panel, and they, interestingly enough, saw a greater benefit for the new antibody. This is the best overall response rate. So it looks like there may be a little improvement. It doesn't look as dramatically better so far in humans as it did in the mouse models, and I think we need to wait for the proper phase 3 trials to end. And there's a whole bunch of phase 3 trials ongoing right now. So we hope that this will be an advance, but I think the jury's still out. The bottom line is rituximab is a terrific product, and it's proving very hard to show head and shoulders improvement over it. The new antibody was thought to was hoped to induce fewer infusion reactions. In fact, it's pretty similar to rituximab in the side effect profile. Here you see any adverse events, most of which were infusion reactions, and the GA101 in blue bars looks pretty similar to the rituximab. A more novel design involves genetic engineering, and again, Jeff Ledbetter, who's here, invented these small modular immunopharmaceuticals, which do not have the CH1 domains in them. They can be expressed in CHO cells, and they spontaneously dimerize. There have been such SNPs made against CD20, and they have been in clinical trials and look good in rheumatologic disorders. For whatever reason, the company has decided to focus its lymphoma work on the CD37 antigen. And this is a tetraspanin molecule, which I won't go into in great detail. And again, in mouse xenograft models, the TRU-016, which is what they're calling the SNP, outperforms the rituximab shown in blue. So these are percent survival of mice with lymphoma xenografts growing, and you can see survival is better with the SNP than with rituximab, and here's the control with no treatment. If you look at the percent tumor-free, it's 75% with the CD37 SNP and 0% with rituximab. So this part looks good. It is in clinical trials, and John Pagel at our institution is a lead investigator on this. And the trial that is being done now is in chronic lymphocytic leukemia, where there are a lot of malignant lymphocytes circulating in the blood. And so if you look at the number of lymphocytes on this axis, and then you look at the initiation of treatment, you can see there's a dramatic disappearance of the malignant circulating lymphocytes from the blood. That's terrific, and in addition, the enlarged lymph nodes that the patient had also went away. Now, if you look at patients overall, so each of these sets of bars is a single patient, and the blue bar shows the lymphocyte count before treatment started. The magenta bars show the lymphocyte count after one cycle of treatment, that is 1 month later. And you can see in virtually every patient, the magenta bar is lower than the blue bar, indicating substantial reduction. So I think the jury is still out. These are ongoing trials, but it looks like CD37 may be another target that can be used either instead of or along with targeting CD20. A third target that looks very attractive is CD22, and there is a humanized antibody called Epratuzumab that's been made, which targets B-cell malignancies. One of the advantages of this antibody, not only does it have a different target, but it can be infused in an hour, which would be much faster than rituximab is usually given. And it is effective at inducing both ADCC and apoptosis. And again, deliberately, the complement fixation was engineered out of the molecule. Whether that's right or wrong remains to be seen. If you look at single-agent trials in relapsed follicular lymphoma, the overall response rate of 43% is about what you would expect in Rituximab trials. Now, these are not head-to-head comparisons. These are separate phase 2 studies, so you need to take them with a grain of salt, but at least it looks like targeting CD22 gives response rates that look somewhat similar to targeting CD20. Now, the use of this is probably not going to be head-to-head competition with Rituximab, but probably using it as a combination. For example, there is a trial using Epratuzumab, Rituximab, and the CHOP chemotherapy regimen, and compared at least to historical controls, it looks somewhat better than our CHOP in aggressive lymphomas. And so there now is a phase 3 trial. This is a historical comparison, so again, there are a lot of flaws in this kind of analysis, but there is a phase 3 trial that's trying to prove that the addition of this antibody will cure more patients than just R-CHOP. Remains to be seen. Another novel approach is being taken by a molecule called Blinatumomab. And what Blinatumomab is a molecular fusion of the single-chain antibodies against CD19. This is a B lymphoma target, and CD3, which is a antigen on T lymphocytes. And so you clone out the single chain from a CD19 antibody and the single chain from a CD3, and you put them together with a small linker, and you wind up with what's called a bispecific T-cell engaging antibody. So what it does is it brings lymphoma cells right next to cytotoxic T lymphocytes. And when that happens, it makes the T cells angry, and they release perforins and granzymes which punch holes into the lymphoma cell or the leukemia cell, because CD19 is also on ALL, it's also on CLL, it's on virtually all B lineage malignancies. And when that happens, you activate caspases and the cell dies. Now, this is a very vigorous response, and there is active release of multiple cytokines, interferon gamma, tumor necrosis factor, a variety of things. And so patients usually get fever when this happens, and they can be quite ill. Matter of fact, if you're not careful and don't pre-medicate with corticosteroids, some of the people actually developed encephalopathy from the cytokine release. And so it needs to be done kind of carefully. That's been reversible in every case. Nevertheless, it is sobering, and there's some tricks to giving this molecule. Nevertheless, it has shown very impressive activity. The overall response rate at the maximally tolerated dose in follicular lymphoma was 83%, mantle cell lymphoma 71%, diffuse large B-cell lymphoma 55%. And so these are all patients who had recurred after getting standard treatment. So they're tough patients, high response rates, and perhaps most impressively, the toughest group of patients to treat are those with relapsed large cell lymphoma. We were getting a third of the patients into complete remission just with this antibody construct. So this is pretty exciting for those of us in the field. It's being targeted particularly for acute lymphoblastic leukemia. And this is a disease that is very curable in children, but when it's not cured, it's hard to salvage, and in adults, it's much, much tougher to cure. And yet, they took 25 patients who were in frank relapse with circulating blast cells and bone marrows full of blasts, and 17 of the 25 achieved not only a morphologic complete remission under the microscope, but if you did PCR, you could not find evidence of the malignant cells by that technique either. And that was true also for some cases that had the Philadelphia chromosome or a translocation between chromosomes 4 and 11. These are particularly bad actors in ALL. And so this was very exciting work. This just shows the relapse-free survival curve for patients with ALL who are treated, and you can see going out several years that about 60% of this small group of patients remains free of recurrence. And so this is an exciting development that is actively being investigated in multiple clinical trials. Another approach to make antibodies work better is to put cytotoxic radionuclides on them. This is an area of work that we have done quite a bit at our center. Ajay Gopal, who's here in the audience, has led much of this work, and John Pagel, a colleague, and many other members of our division have worked on these studies as well. If you attach a radionuclide to an antibody and target it to tumor cells, you can kill those cells by emission of beta particles, and with radio-labeled antibodies, you get a crossfire effect that you don't see with naked antibodies or with antibody-drug conjugates or immunotoxins, because the beta particles travel over several cell diameters. And so if you have a cell in the center of a clump that doesn't express the tumor antigen, that is an antigen-negative variant, or a cell that just doesn't have access to the antibody because the antibody hasn't penetrated well enough, you can nevertheless kill that cell by crossfire. Obviously, there's a trade-off. If there's a normal cell on the periphery, it can see a beta particle whizzing by too. So there's a little give and take that happens with these. But in a head-to-head randomized study, using a radio-labeled antibody called Yttrium-90 Epratuzumab Tiuxetan, the FDA is wonderful how they give these names, and this is the mouse monoclonal from which Rituximab was engineered. So this is a very good comparison. You can see that the radio-labeled antibody outperformed Rituximab head-to-head with an overall response rate of 80% versus 56%. The complete remission rate was 30% versus 16%. So this looks like a better product. Admittedly, there was a trade-off in that there were some cytopenias with the radio-labeled antibody. They were temporary and well tolerated, but you don't see those with Rituximab. And so, like most things in life, there are advantages and disadvantages. We and others have done studies in which we tried to incorporate radio-labeled antibodies into frontline treatment. And here, the complete remission rate is in red, the partial remission rate is in green, and if you combine radio-labeled antibodies with a variety of upfront chemotherapy regimens, you routinely get overall response rates between 90 and 100% in every trial, and you get complete remission rates between 60 and 95% depending on the trial. So this was very exciting. We did a phase 2 study in the Southwest Oncology group, with now a 10-year follow-up of that study, we still have 60% of people who are continuously progression-free after 10 years, and 80% who are alive. So this looked like a very promising treatment, and we went on in the Southwest Oncology group to a randomized comparison. It was designed many years ago, and if we were doing it today, we would design it somewhat differently, but the comparison that was made was CHOP, which is given for 6 cycles with 6 doses of Rituximab, against 6 cycles of CHOP with no Rituximab and a single dose of a radio-labeled antibody, which was iodine-131 Tosatumumab. And there was a trend towards a better progression-free survival with the CHOP-RIT regimen, but it was not statistically significant to our chagrin. And there also wasn't an improvement of overall survival. In retrospect, what we wish we had done is R-CHOP with or without the radio-labeled antibody rather than a head-to-head comparison. So, but hindsight is always 20/20. This was designed in 1999 before Rituximab was the standard of care, and it's hard to see. These studies in indolent lymphoma take 10 years or more to do because the patients live a long time. Which is a good thing. Perhaps more compelling is this randomized trial from Europe in which patients who were treated with any type of chemotherapy, once they achieved a partial or complete remission, were then randomized to either get a single dose of an yttrium-labeled CD20 antibody or no further treatment. And in this trial, there was a statistically significant improvement in the progression-free survival. It was 4 times greater, 49 months versus 12 months in those that didn't get consolidated with the radio-labeled antibody. So this is a quite compelling trial. The updated version of it is online. It's not yet out in print form. In our laboratory, Jay, John, and I, and others have looked at new ways of trying to do this better. And I've talked to you so far about just directly radio-labeled antibodies. but there is a multi-step method of separating the delivery of the radionuclide from the antibody using an adapter molecule of streptavidin or other adapters can be used. And then after maximal accumulation of the antibody conjugate in the tumor, you then give the small radionuclide, which zips in on biotin and binds essentially irreversibly to the tumor-bound antibody. And you get an amplification effect, and you also get rapid excretion of any unbound radionuclide. And this works very well in mice. Here you see mice with human lymphomas implanted in each flank. And if you do imaging with a gamma camera, you can find that with conventional single-step radioimmunotherapy, you get nice imaging of the tumor, but you've got a lot of radioactivity in the blood pool of the mouse for the first 24 hours, and you see some in the urinary bladder, whereas with pre-targeting, all the activity really is in the tumor. So that's what you'd like. It also cures the mice much better. Here you see with the pre-targeted method, we can cure virtually all the mice who are bearing a Burkitt's lymphoma xenograft. Whereas with conventional radioimmunotherapy, we get a response, but then a relapse in this very aggressive model. And these are two control groups. So this is very exciting. We have a clinical trial that's opening now with radiolabeled antibodies. The first trial is being done by John Pagel in the myeloid leukemia setting, but we also have one planned that Ajay will do in the lymphoma setting. A new and very exciting area is the field of antibody-drug conjugates. In this area, we're, instead of giving the chemotherapy as a separate infusion from the antibody, as we do with R-CHOP or RCVP, here we're actually attaching the cytotoxic chemotherapy drug right onto the antibody and focusing it specifically on the tumor cells. And there are a variety of these that have been made. This one called Rituximab vedotin is now FDA approved. It targets a molecule called CD30 on the cell surface. And an important point for the antibody-drug conjugates is you need to target an antigen that's going to be internalized rapidly into the cell, because you need to get the drug inside the cell, which you don't with a radionuclide, and you don't want that to happen with rituximab or a naked antibody. You want it to be on the surface where it can interact with complement or effector cells. But here you need to get it inside the cell. So you don't want to target CD20, you want to target something that goes inside the cell like CD30. And here you can see that it's internalized. The drugs in the acidic environment of the endosome or lysosome are released. And the particular drug that's used here, monomethyl auristatin E, is a tubulin inhibitor, and so it kills the cells when they try to divide. And this drug, which used to be called SGN-35 because it's made by Seattle Genetics, has been tested at our center by, again, Ajay Gopal and Andrei Shustov. And you can see that in patients who had failed prior treatments, including an autologous stem cell transplant, nevertheless, nearly all the patients had tumor shrinkage, as shown by bars going below the line. Only a couple of patients progressed through it. And if you look at objective remissions, 75% had at least 50% shrinkage, and a third of the patients had complete remissions after failing conventional chemotherapy and after failing an autologous stem cell transplant. So a very exciting drug, got it FDA approved. Equally exciting results were seen with a kind of T-cell lymphoma that expresses CD30 called anaplastic large cell lymphoma. And here you see that 97% of patients with this type of lymphoma responded to this antibody-drug conjugate, and all of them had failed prior CHOP chemotherapy. So this is an exciting drug. Half the patients got complete remissions, many of which have lasted long-term for more than a year or two so far and are still in remission. So this is a very exciting drug, and there are efforts to incorporate it now into frontline treatment. It may become part of the standard treatment for all patients with Hodgkin lymphoma. It isn't now, but it may be in the future. Trials are ongoing. The problem with targeting CD30 is it's not on most of our lymphomas and leukemias in very great degrees. It's on Hodgkin's and anaplastic large cell lymphoma, but the vast majority of lymphomas don't express a lot of CD30. So it'd be nice to have these antibody-drug conjugates against more commonly expressed antigens. And there are some widely expressed internalizing B-cell antigens, CD22, CD19, CD79. CD79 is the signaling portion of the surface immunoglobulin that's on every B cell. And you can make an antibody against this molecule. If you do that and test it in a mouse lymphoma xenograft, you can easily cure at even 2 well-tolerated doses. You can cure every mouse that is bearing a tumor. Even when R-CHOP chemotherapy doesn't do it, Rituxan alone doesn't do it in red, and so this, again, in mouse models looks great. And there is an ongoing clinical trial. It's a phase 1/2 clinical trial. It's actually recently been completed. Karina Polanka-Wessels in our group was the lead investigator and presented the results at our hematology meetings in December. And it's been done in a variety of histologies. But some of the responses have been dramatic. This is a patient we treated at the SCCA who had a mantle cell lymphoma. This is all lymphoma in the armpit of the patient. Before treatment and after 3 cycles of treatment, the lymphoma is essentially gone. So very dramatic disappearance. If you look at the waterfall plots, results are immature because a lot of the patients are still in remission and are still on treatment having tumors that are still shrinking. And so we don't know yet the ultimate extent of complete response. But what we can say on the waterfall plot is that most of the patients with a variety of different types of lymphomas are experiencing gratifying shrinkage. And that includes very tough lymphomas like transformed follicular lymphoma, diffuse large B-cell lymphoma, mantle cell lymphomas. So this is, again, a very exciting avenue to explore. CD22 is also an excellent internalizing target for these agents, and there are several companies that have produced CD22 antibody drug conjugates. This is one called Inotuzumab azogomycin, and it involves a CD22 antibody that has a linker connecting it to this very potent chemotherapy drug called calicheamicin. And in results that were just published a month or so ago, you can see that in patients with relapsed follicular lymphoma who had failed conventional rituximab and chemotherapy treatments, that virtually all the patients had tumor shrinkage, and 87% overall response rate, 62% complete remissions. Very exciting. More exciting is the results in aggressive lymphoma. Although the numbers aren't quite as good, this is a much tougher lymphoma. A relapsed diffuse large B-cell lymphoma that has not responded to R-CHOP or recurred after R-CHOP. Many of these patients had had transplants already. And when they were treated with this single agent, 74% had objective remissions and 50% complete remission. So this is a really tough group of patients, and for us is very encouraging. If you look at the progression-free survival curves for follicular lymphoma and diffuse large B-cell lymphoma, you can see that many of these patients are going out several years and remaining both alive and free of tumor progression. There is a group of patients who are refractory to everything who were treated with this, and some of them responded, but those patients had very short remissions, unfortunately. The final approach that I'm going to talk about is adoptive T-cell immunotherapy. This is a huge field. I just gave an hour-long talk on this a week ago, so I don't have an hour left. So this is going to be a bird's-eye view, and again, I apologize. There are people who will feel I haven't done justice to their hard work, and I'm sorry, I just don't have time to do that. As much as I've been lauding antibodies, and most of my career has been spent doing antibody-related research, most tumor immunologists will tell you that in a native organism, usually the cellular immune system is more important than the humoral immune system. That is, T cells are probably more important in keeping us from getting cancers than antibodies. But they've been tougher to harness than antibodies. Many of you know that cytotoxic T lymphocytes recognize tumor cells and other things in the form of digested peptide antigen, which is presented in the context of the major histocompatibility complex. And in order to get good T cell function, good killing of a target cell, you need to have at least 2 signals. you need to have the T cell receptor engaging peptide antigen in the context of MHC, but you also need to get a second signal. And that second signal can come from a variety of different partners. So there's a molecule on T cells called CD28, which binds to either CD80 or 86 on an antigen-presenting cell, and that can give a signal. Or there is a molecule called 41BB or CD137, which engages a partner on antigen-presenting cells, and you need one or the other or both of these to get an effective signal. If you just get a single signal, T cells often become anergized, where they actually don't respond to the target, or else they undergo apoptosis, where they just commit suicide and die. So you need the second signal usually to make it work. And the basic concept is shown here in a slide I stole from Mike Jensen, who's at Children's. And the idea is that you draw blood out of a patient, you take out their T cells, and then you genetically engineer those cells so they do what they haven't been able to do in the patient's body. That is, you give it a molecule that makes it recognize the tumor and kill it more efficiently. You grow up billions of those cells in your laboratory, and then you reinfuse them into patients. One of the problems is that it takes a little time to grow up these billions of cells. You can see here the guy looks like he's 6, and here he's 25. That doesn't really take that long, but initially it took us 3 or 4 months to do this work. Brian Till in the lab and Elizabeth Buddy and Chandra Bruns and others are working away on this in collaboration with Stan Riddell, who's a world leader on this field, Phil Greenberg at the Hutch, Mike Jensen at Children's now. And but initially it took us 3 or 4 months to raise up these cells. Now we've got it down to about 3 weeks, but it still does take some time to do it. And this is, of course, a form of gene therapy, and I'm a little worried with the changes in the healthcare environment, whether we'll be able to do the kind of gene therapy I'm talking about, or whether this will be the kind of gene therapy we're doing. But the basic idea is you can either try to transfer a native T cell receptor that recognizes a tumor cell, or you can synthesize your own artificial or chimeric T cell receptor. And this is what most people are doing. What it involves is making a new T cell receptor that recognizes antigen like an antibody would. You actually clone out the single chain region of an antibody, and then you fuse it to a signaling domain that turns on the T cell like a T cell receptor would. And most people have borrowed the CD3 zeta chain from the T cell receptor. That's what we've done and what Stan Riddell and others have done. You can also instill some of the co-stimulatory domains, CD28 and 41BB. You can put those in here, and it turns out they don't have to be on separate molecules. You can engage and make this work in a single molecule. And Carl June, who was a fellow with me here at the UW and the Fred Hutch 30 years ago, is one of the world leaders in this field now. He's at the University of Pennsylvania. But he views these chimeric antigen receptors, or CARs, in several generations. You have the first generation with just the single chain and the CD3 zeta. Then you have a second generation that has the single chain, CD3 zeta, and either CD28 or 41BB. A third generation CAR, or a muscle CAR, would be with all of those domains. And so you've got classic CARs, sports CARs, and muscle CARs. And if you do this and target pediatric diseases, you also have pediatric CARs. So I thank Carl for that slide. I think that we are in this position with adoptive T cell therapy. We're pretty sure that this is going to be a really important avenue. We're not sure that we're using it the best way right now, but we're kind of going through the early stages of experimentation. And we have, Brian Till in our group has done 2 trials using CD20 CARs. That is, they recognize the CD20 antigen just like Rituximab does, but then we're signaling T cells through this construct. The first one was a first generation CAR that just had CD3 zeta, and we got this construct from Mike Jensen, who at that time was City of Hope. And then we modified it for the the last group of patients by adding CD28 and 41BB co-stimulatory domains as well as a translation enhancing domain. I'm not going to go into the detail, but this is a picture of our T cells growing in our flasks in the lab. And Brian has done a couple of trials that basically involved enrolling the patient, having them undergo apheresis to collect T cells, and then while we were transfecting them, we initially used electroporation and growing them up. Patients could get interim chemotherapy if they needed it. We then often gave a dose of cyclophosphamide to get rid of the normal T cells and to create space for the adoptive transfer of T cells so that they would have room to grow and would have the cytokine milieu they needed to grow optimally. And then we did 3 escalating infusions of T cells. And in some of the patients, we also gave subcutaneous interleukin-2 as a T cell stimulating factor. So I don't have time to go through the details of the trial. I will say that all the trials in this field are boutique trials. There was no trial done by any investigator yet which has more than 10 patients, at least in the published trials. And so it's early days. We don't know exactly where this is going. This is the first patient that we treated years ago at the CRC here at the UW. And Brian Tilla's got a couple of reports in blood on this. It's, at this point, still a fairly laborious process. He has enrolled 14 patients, 10 of whom have actually received the infusions. I'm not going to go through all the findings. The cells with the co-stimulatory domains definitely grew faster and killed better. And we did see some clinical responses. We saw a complete remission, a couple partial remissions. But there's a lot of room for improvement. We need to improve our gene transfer efficiency. We need to improve the expression of the CAR on the cell surface, the persistence of the T cells in the blood. And this just shows 3 groups that Brian treated. Here you see where we did single cell cloning. That was very laborious, and a lot of the cells were exhausted by the time we gave them to the patient. They didn't last very long in the bloodstream, only a few weeks. Here, if you look at persistent of CAR-expressing cells, in those grown with bulk culture with interleukin-2, you can see we keep them around for about 3 months. And in the last group of patients, where they had the co-stimulatory domains as well as IL-2 in bulk culture, we're able to detect low numbers of cells out for a year or so. So that's good, but I think we still need to make a better milkshake. And so Elizabeth Buddy in the lab has engineered a lentiviral vector that incorporates the CD20 CAR gene, but also incorporates a suicide gene in case the T cells create bad things like permanent B cell ablation would be bad, or leukemogenesis. It's possible when you're doing gene transfer to create insertional mutagenesis. We also put in a selection marker, a truncated CD19 molecule, which is usually not on T cells. And I'm not going to go through this, but we can induce at will apoptosis using a dimerizing molecule which brings together this fusion protein that incorporates inducible caspase 9. This just shows some flow cytometry of T cells that we have transduced with a lentiviral vector. This is work by Elizabeth Buddy that's been submitted for publication. And in this 2-color analysis here, the CAR expression goes in this direction. The expression of the transferred truncated CD19 molecule goes in this direction. You can see in the mock transduced cells, there are no cells in this quadrant, whereas about 80% are here. So this is a very efficient process with very high expressing CARs. In a mouse model with luciferase transduced Burkitt lymphoma cells in the mice, with mock T cells, you can see that the tumor grows rapidly. By 19 days, they're multicolored, showing the tumor growth, whereas with the transduced T cells, we were able to cure most of the mice. An occasional one still had a small amount of tumor. And if you look at survival of the treated mice with the adoptive CARs, about 70 or 80% were cured, whereas in the control group, they all died. So we are, this is my wife with me at my doctor's appointment, and she says, if the medicine worked on rats, it'll work on Ollie. And we're at that point in the development where we're trying to take this new CAR that Elizabeth's produced with the help of Stan Riddell and Mike Jensen and others, into the clinic, unfortunately, there are a lot of regulatory things. So we're trying to make the vector GMP. We're trying to get over all these hurdles, but it's going to take us a year or more to do that, unfortunately. Now, obviously, there are many other groups involved, including Stan Riddell at the Hutch, who has a CD19 project. Our two publications are in yellow, you can see. There are groups at Sloan Kettering and elsewhere. The targets are mainly either CD19 or CD20. Most people are choosing CD19. And perhaps the person who's gotten the most publicity here is Carl June, who's used the CD19 CAR and a 4-1BB co-stimulatory domain. In his hands, that leads to the best cell growth, as shown in this line. And again, I have to skip over this very lightly, but he's treated, I guess, 12 patients, 10 of whom had chronic lymphocytic leukemia and 2 of whom had pediatric ALL. He's had some complete remissions, very dramatic complete remissions, some of which have lasted more than 2 years. And these are in patients who had failed multiple treatments. So these are very encouraging results. When this works, he gets total B-cell aplasia. Here are patients before treated. You can see the B cells are in this quadrant. After treatment with the CAR transduced T cells, he has no B cells. Not the malignant B cells, not the normal B cells, no B cells. And a year and a half later, that's still the case. Now, some of these patients required supplementation with intravenous gamma globulin. You can do that. With our construct, if this becomes a problem and we think we've cured the patient, we could activate our suicide gene, kill the T cells, and let the normal B cells come back from the pluripotent stem cells. So that's kind of the concept and the hope. And there are just 2 patients that he's treated with CLL who have ablated their B cells. So it seems to be very effective. You do get B-cell aplasia, and hypogammaglobulinemia can be an issue. And this is very potent treatment. Some of the patients get tumor lysis syndrome. Some of them get life-threatening cytokine release syndrome with interleukin-6. And some of them have wound up in the ICU on ventilators. They then got complete remissions, and it does appear that you need to have a relatively potent interaction to get this effect. And it is reversible. It turns out that an anti-IL-6 receptor antibody called Tocilizumab can make this very high fever go away almost instantaneously and reverse the cytokine release syndrome. So it is a potent treatment, but we very much are feeling it out, and it can be quite toxic. At the Hutch, we have 2 active trials at the moment. My trial is not active, but Stan Riddell, Cameron Turtle, and Mike Jensen have a trial. They are actually using immunomagnetic selection to select out central memory T cells. So rather than transducing all T cells, which is what we've done in my lab, they are, have a more sophisticated way where they are first isolating central memory T cells, transducing them, and then infusing controlled numbers of central memory CD8 cells along with controlled numbers of CD4 cells. And they are doing this with a CAR that's targeting CD19. And this just shows their lentiviral vector. And as I mentioned, they have several different dose levels and mixtures of CD8 and CD4 cells. And their trial is enrolling. They have another trial for allogeneic transplant patients that's a little more complicated to explain. And since I only have 2 minutes for questions, I think I won't have time to do that. I do have to say that much of what I've told you today is in the early stage of development with phase 1 and 2 trials. We do need to have a bigger phase 2 experience with the CAR T cells for sure, and ultimately the value of all these things will need to be shown in randomized studies as shown here. And I will end by acknowledging that the people in my group is shown here, particularly Jay Gopal, John Pagel, who've worked with me for many years. They're both associate professors in their own right, Damian Green and Brian Till, assistant professors, Corinna Polanco-Wessel is an acting assistant professor, and then all the other fellows, nurses, technicians in the lab, without whom none of this would be possible. Not to mention my wife, who keeps us all in line. And Stan Riddell, Mike Jensen, Carolina Berger, David Spencer have all been invaluable in allowing us to do the CAR T-cell work I mentioned. I'll have to thank the NIH, which has been good to us so far, and the Giuliani and Edson Foundations, who have also supported our work. So with that, I'll wish you all a good day, and I think I've used up all my time, but I'd be happy to answer a question or two. Should I do that down front, or? Yeah, I'll just take questions down front if you got a question.